Hi, my name is Christina Yang, and I am in the laboratory of Karen Ottoman at the University of California, Santa Cruz, in the Department of Microbiology and Environmental Toxicology. And today I'm going to be talking to you about how this pathogen called Helicobacter pylori uses chemotaxis to modulate host inflammation. So the gastrointestinal tract harbors trillions of microbes that are collectively referred to as the gut microbiota. And most of these microbes are actually harmless or even beneficial to our health, but there are some that are pathogenic. So an example of a pathogen that is a part of our microbiome is called Helicobacter pylori. And H. pylori is a chronic pathogen that colonizes the stomach. This is a long-term infection that causes chronic inflammation, which is asymptomatic in most individuals, but a subset of the infected individuals will go on to develop gastric ulcers or even gastric cancer. But it's not well understood why only a subset of those infected develop severe pathologies while others remain asymptomatic. So one factor that seems to drive the pathology is severe inflammation. So patients with higher inflammation caused by H. pylori seem to have a higher incidence of developing gastric ulcers or gastric cancer. So we're really interested in understanding factors that contribute to inflammation. So this is interesting to us. So we saw that H. pylori, if you knock out this gene called TLPB, which encodes for something called a chemoreceptor, you see that these mutants cause higher levels of inflammation compared to the wild type. And this is independent of the total number of bacteria that are colonizing the stomach. So the mechanism by which these TLPB mutants cause higher inflammation is unclear. So chemotaxis is used by bacteria to sense and respond to chemical stimuli in its niche. So a TLPB is a chemoreceptor that H. pylori senses and responds to its chemical stimuli. And when something favorable called an attractant is sensed, H. pylori will swim towards the attractant. But when something bad or a repellent is sensed, it swims away from it. So this is really important in the stomach environment because there are three distinct microenvironments in the stomach. You have the acidic lumen area, you have the mucus area, and you have the gastric glands. So H. pylori is known to colonize the mucus layer and the gastric gland area. And the population in the gastric gland is particularly interesting to us because it is in close proximity to the lamina propria, which houses the immune cells. So we wondered what the localization of the TLPB mutants were when we infected our mouse model with it. So what we saw was when we infected mice with TLPB mutants, they actually have a larger population of bacteria that are residing in the gastric glands compared to mice that are infected with wild type H. pylori. And we saw that there was the same total number of bacteria. So we really think what is happening is that H. pylori is using chemotaxis to regulate the number of bacteria in the gastric glands. And this is used to modulate the amount of inflammation that is elicited by the host to control the infection.